Welcome back to the STS Biology Review Pod. I'm Schuyler from Seattle Tutoring Services.
And I'm Leslie. Today we're continuing our foundational journey through AP Biology, zooming in on how life's chemistry actually works at the molecular level. Last episode, we explored the cast of chemical elements found in living things, from the carbon in your DNA to the iron in your blood. But knowing which elements are involved is only half the story. The real magic happens when these elements interact, especially in water. When elements and compounds dissolve in water, they often break apart into ions. These are particles with a positive or negative charge. In your body, for example, sodium, potassium, and chloride ions are everywhere. They help with nerve signaling, fluid balance, and pH regulation. And when these individual elements interact in water, their chemical behavior can change dramatically compared to their behavior in compounds. Now let's see how this leads us to acids and bases. Recall that a typical hydrogen atom has a proton in the nucleus, but no neutron. Surrounding its nucleus is just one electron. An acid is any substance that donates a hydrogen ion. In other words, the nucleus, which is just a proton, separates from the surrounding electron and enters a solution by itself. Therefore, you may hear people refer to a hydrogen ion simply as a proton. Let me say this again because it's so important. If a proton leaves a hydrogen atom, that is an acid. A base either accepts a hydrogen ion or donates a hydroxide ion. A hydroxide ion is negatively charged, so in the presence of a proton and its positive charge, it will combine to make water. If you add something that increases the proton concentration, it's an acid. If you increase the hydroxide concentration, it's a base. Vinegar, for instance, is acidic because acetic acid releases a hydrogen ion and changes H2O to H3O. Here's an example of a base being combined with water. Sodium hydroxide dissociates into sodium ions and hydroxide ions. To measure just how acidic or basic something is, we use the pH scale. It ranges from 0 to 14 with pure water sitting in the middle at 7. 0 is the most acidic possible, and 14 is the most basic possible. The pH scale is logarithmic, meaning each change by one number represents a tenfold change in proton concentration. For example, a liquid at pH 5 is 10 times more acidic than a liquid at pH 6. Now, why does pH matter for life? Most living things operate in a narrow pH range, usually close to neutral. Your blood's pH needs to stay between 7.35 and 7.45. If it drifts even a small amount higher or lower, it's dangerous. Stomach acid is about a pH of 2, essential for digesting food. A cell's cytoplasm is nearly neutral, protecting delicate molecular machinery. The structure and function of proteins, including enzymes, depend heavily on pH. If the pH is off, proteins can lose their shape and can't do their job. This can cause cells to malfunction, or even cause death. In humans, pH imbalances called acidosis or alkalosis can be life-threatening. In the environment, things like acid rain lower the pH of lakes and rivers, putting entire ecosystems at risk. So how do living things keep their pH in the safe zone? That's where buffers come in. A buffer is a substance that minimizes pH changes by soaking up or releasing hydrogen ions or hydroxide ions as needed. Buffers are chemical shock absorbers, helping to keep the environment stable for life's chemistry. A great example is the bicarbonate buffer system in your blood. It balances carbonic acid and bicarbonate ions to keep blood pH steady, even when you're breathing hard or exercising. Similarly, phosphate buffers inside your cells help maintain the right pH for all the complex reactions happening every second. You'll encounter buffers in laboratory experiments, like enzyme labs, or even in food science, where pH control prevents spoilage. Now, you might be wondering, why spend so much time on acids and bases before talking about the big molecules, like proteins and DNA? Proteins and enzymes, for example, will unfold and become inactive if the pH strays too far from normal. The shape and stability of nucleic acids, cell membranes, and even how signals travel from cell to cell are all pH dependent. Think of it this way: you can't build a house without understanding how solid the ground is. In biology, you can't study macromolecules without first understanding the chemistry of their environment. So, now that you've got the basics, elements, the significance of water, ions, acids, bases, pH, and buffers, you're ready to see how life's most important molecules are built and maintained. In our next episode, we'll start unpacking the world of biological macromolecules, carbohydrates, lipids, proteins, and nucleic acids, and you'll see exactly how all these chemical rules come together to give life its structure and function. Here's a question to think about for next time. Why does a fever make you feel so awful? And how can your stomach digest protein but not everything you eat? Stick around to find out how life's biggest and most important molecules depend on the chemical principles we covered today. Quick reminder, if you want extra help acing AP Bio, join our weekly interactive review sessions. It's $65 per person, but if you organize a group with friends, it's just half price each. See the show notes or go to seattletutoringservices.com or text 206-999-0666 to register.